Okay, for the chemical digestion, if you remember, we said we have several places for it. We have it in the mouth. We have very limited amount of chemical digestion there. Then we have a stomach because of the HCl, and the main part or the main chemical digestion will happen in the small intestine. Some of it will happen also in the large intestine, but the main part will happen in the small intestine. Now, for whatever material we are consuming, whether they were like sugars or carbohydrate or proteins, those need to be broken down into their monomers. What does it mean? Into their building blocks. Those building blocks for the carbohydrates are the simple sugar. Like what? Like the glucose, for example, like fructose, lactose. All of those are simple sugars. Proteins, the building blocks of them, if you remember, is the amino acids. The point of the chemical digestion is to get the proteins into their into their amino acids, and to get the carbohydrates into their simplest form, into the simple sugar. Only those what the cells can absorb. Okay, not the disaccharides, not the dipeptide, not the polypeptide. No, the simplest form of them. So for the carbohydrate digestion, mainly we are talking about the starch and glycogen. Those are the polysaccharides. This is how we usually eat them, like from plants or from animals. In the saliva, we have the salivary amylase that will cause some digestion to the starch and glycogen, but and sometimes it will be like enough to break them into disaccharides, but in most of the time those would go to the duodenum. Okay, in the duodenum also we will have the pancreatic enzymes, mainly pancreatic amylase. This pancreatic amylase will break down the starch and glycogen into disaccharides, and then in the small intestine we have the brush border enzymes. Those will turn the disaccharides into monosaccharides. So we have the lactase will turn lactose into glucose, galactose. Saccharase will turn sucrose into glucose and fructose. Maltase will turn maltose into glucose. And we have the alpha dextrinase will turn the dextrins into glucose. Okay, you need to be familiar with those. Okay, just the names at least: glucose, galactose, fructose. You need to be familiar with those monosaccharides. For the proteins will pass through the mouth all the way to the stomach, and that is where the start of protein digestion will occur. Why? Because mainly of the HCl will denature the proteins, so it will cause them to lose their 3D structure. So now we will get them into a sequence of amino acids. The pepsin from the stomach will start to break those chains of amino acids, and this will progress into the duodenum. From the duodenum, we said we have the pancreatic juice that will secrete trypsin, chymotrypsin. Those also will further break down those chains of amino acids into single amino acids, and that is what will get absorbed by the cells, the single amino acids. So the most important part is to understand that proteins will pass to the stomach almost intact. There we have the HCl, we have the pepsin, and then in the small intestine we have trypsin, chymotrypsin, and will break down to the level of amino acids. For the lipids almost they are not really digested. We absorb lipids as they are, but the main thing about them that they need to be emulsified. Remember, those are like fatty acids and monoglycerides. We don't dissolve them into smaller units because we will use them as they are. We will use them to build our cells. But the point is we need to emulsify them, and that is where the bile will come. The bile will act like a soap on if you have a dish that is filled with grease, you will add some soap and it will break down the fat, right? So this is kind of the same concept. Emulsification will cause breakage of those fatty acids and monoglycerides in smaller units. Those eventually we call them the cells, and this is what being reabsorbed through the cells and processed to the lymphatic vessels or lymphatic capillaries, and from there they will enter into the circulation. So they pass almost intact, and we will reuse them in different parts of the body. Okay, for the lipids, the point is that when we consume fat, we will have the lipases. Lipases are the enzymes that will break lipids. But those lipases from either the saliva or the salivary gland or the stomach, they are not really effective. It's only the pancreatic lipases. Those are the ones that would break the triglycerides. Okay, the triglycerides will get broken into fatty acids and the monoglycerides, and those will get emulsified and absorbed by the intestinal cells. So emulsification, again, we need to put them in a layer of bile that would make them hydrophilic, and then attach them to the microvilli, and this will allow them to get absorbed or technically passed through the cells into the lymphatic vessels, because they are too big to go into the blood. So they will be passed into the lymphatic vessels, and from there they will get into the circulation. But the point is we need to emulsify them. Okay, then the last thing is the nucleic acid digestion. Nucleic acid in general, they are not really a good source of energy. Okay, but since we are eating cells, we are eating also nucleic acids, so those will get digested, you know, by the pancreas enzymes into their singular components, but again, and will be reused to produce, you know, or to make DNA and RNA in our body, but they are not really a source of energy. Okay, so that's it for this chapter. Let me know if you have any question.